Well, good morning, good afternoon, and I believe we have a good evening participant on as well. Welcome to Innovative Health's second virtual learning session in place of HRS 2020, which has been canceled this year. If you missed Monday's first virtual HRS session from Innovative Health, you can find and access a video of that on the landing page um, that launches our virtual HRS program. Today, our session is going to be scientifically focused. We're going to be talking about lumen catheters and how to clean lumen catheters. And the reason why it's important, there's a couple of reasons why. One is that the more expensive devices that are used in the EP lab have lumens in them, and quite a few of them increasingly have microlumens in them as well. Historically, reprocessors have not approached such devices. They have been considered not possible to clean. And the history of reprocessing lumen catheters altogether is not very long. The reprocessing of microlumen catheters is very, very new. And that's what we're going to be focusing on today. It's a reprocessing technology that Innovative Health has developed as the only reprocessor in the country. Before we get started, a few introductions. Today's presentation is going to be by Aaron Fowler, who is a senior engineer with Innovative Health and the leader of our emerging technology team. Rafael Chutzek, who is the VP of R&D and Operations at Innovative Health, and Blessing Joseph, who is the Director of R&D with Innovative Health as well. For courtesy, please mute yourself unless you're asking questions. There will be places during the presentation where the presenters will stop and give everybody a chance to ask. Before we get started and get to the more interesting pieces in this, let's talk a little bit about reprocessing and where reprocessing is today as a key practice in the use of medical technology. Reprocessing single-use devices is not just a matter of buying cheap devices. It really is a matter of getting the full utilization out of each asset that the hospital purchases, making sure that full value is used up before discarding the device is purchased. There are three pillars in reprocessing as a fairly advanced industry today. Number one, medical device reprocessors can only sell back single-use devices that have been reprocessed if they have a 510K clearance from FDA, similar to original manufacturers, but there are added demands to reprocessors in demonstrating that this can be done safely and without changing the functionality of the device. This has meant that companies that today are single-use device reprocessors organized in the Association of Medical Device Reprocessors, AMDR, are very, very sophisticated companies that function under tight FDA scrutiny. And then note as the last pillar here, the meaning of a 510K clearance from FDA is that the device is functionally substantially equivalent to a new device, and also that the use of a reprocessed single-use device does not imply any added risk to the patient. So as we're looking at reprocessing today, most US hospitals and EP labs take advantage of reprocessing to some extent, but most EP labs, specifically electrophysiology labs, which we're focused on in our industry, do not reprocess as much as they could. There is a lot of opportunity to increase savings from such programs. That is made possible primarily by specialty reprocessors, which is what we discussed on Monday, such as Innovative Health, that has a strong clinical, scientific, and program management focus so that those savings can be maximized. And of course, the relevance of this particular conversation today, again, lumen catheters, advanced, very expensive catheters, are fairly new to reprocessing. Innovative Health wanted to show a little bit of our magic, explain how that is done. Also because questions about cleaning and testing and inspecting very advanced devices is something that is often brought up as questions by clinicians in EP labs. But now more than ever, with the COVID-19 crisis, there's a lot of awareness on the need of EP labs to add more savings, and lumen catheters can play a major role in that. So our focus, and what Rafael and Les are going to go into, is to, as much as possible, keep pushing the bar and obtain FDA clearances to safely reprocess devices that can increase the savings for EP labs. So the difference here, those of you that saw the presentation on Monday have seen this slide before. The difference here from what we used to think was EP device reprocessing as this type of devices. These are quads, fixed catheters, steerable catheters, not terribly complex. These have been reprocessed for many, many years, and it doesn't take advanced reprocessing technology to be able to sell these devices. However, when it comes to devices like these, Innovative Health has FDA clearance to reprocess 2 of them, one in the middle and one to the bottom left. These are devices that can be reprocessed when specialty reprocessors develop reprocessing technology that can handle more challenging devices, such as devices with microlumens in them. So in other words, EP labs can choose not only a discount device program with the easy to reprocess catheters, but actually access the latest technology devices as well, with savings through reprocessing. And I'm going to hand it over at this point to, I believe, Rafael.
Yeah, thanks, Lars. Good morning, everyone. Thanks for taking time today and sitting through this. You know, our hope was that we can provide some education, some insight as to how we clean lumens and our patented lumen cleaning process. As we move into this new platform of lumen devices, we wanted to really make sure that the education was there. We felt that it was important that we pull back the curtain a little bit into how we came across this method, what we did, some of the trial and error that occurred, and then ultimately how that translated into our process. So we hope that you get some education out of this. When the engineering team first looked at this concept, we knew that we had a challenge on our hands that hadn't been tackled really before. There was no solution in our industry to date dealing with lumens this small. So we pulled the team together and really wanted to understand, how do you get a lumen to be clean but also clear? We knew that we've developed some strong competencies with our 32 510Ks, cleaning devices and cleaning external surfaces. That became a solid process for us that's very developed in our production floor. But really getting down into the lumen into such a small space and still being able to show the endpoints for cleaning down to the microgram per square centimeter proved to be challenging. So there was an evolution of the process. There was a lot of back and forth between FDA on the method, and ultimately we were able to find a solution. So again, what we're looking for is to really have everybody understand, and all questions are welcome, to know how these lumens are cleaned and what we have to do to clean them. In terms of the slides, we've kind of broken it down a little bit. As Aaron takes over, we're going to look at some of the initial conceptual principles of fluid dynamics. You have to understand what the water is doing, the physical properties, how is water and the flow of water actually affecting the cleaning of the lumen. Once we understand that, we'll jump into how do you connect the fluid dynamics into a cleaning process. There are very specific parts of the cleaning process that have to be in place to get to a surface area that small, that tight. So Aaron will walk us through how we combine fluid dynamics with cleaning principles and the use of chemicals, temperatures, and other parameters. And then finally, to bring it home, once you understand those two concepts, all that has to translate into an effective process. And so what we've done is we want to pull back the curtain, like I mentioned, and really give you an understanding as to some of the high-level methods of how we used to clean them, but also more importantly, what we do to quality control these devices. We realize that there are things with these devices, lumens this small, that you have to be very careful with in terms of safety for the patient and the user. So we want to talk a little bit about what that looks like on our production floor.
So it really should come as no surprise that some of the most expensive EP devices used today contain lumens, given their complicated and advanced geometries and functions. Typically, a lumen delivers heparinized saline during a study, preventing thrombus formation, but they also serve other purposes. During use, blood and other contaminants may ingress, which need to be removed during reprocessing. At Innovative Health, we have developed several new cleaning and inspection techniques, expanding our portfolio of savings opportunities. ANSI 79 has guided central reprocessing of reusable devices for years. From forceps to endoscopes, these methods have generally been effective for these types of devices. However, one size doesn't fit all, and the same methods and standards simply don't apply to single-use catheters, especially those with a lumen. This is because there's a wide variety of lumens among single-use devices. Those shown here represent only a few of the many available today, and only a few of those we have developed reprocessing methods for. The lumens in these devices are made of different materials, have different specifications like length, diameter, or curve, and may be intended to be used differently. A lumen that is exposed to a transeptal needle must be handled quite differently than the lumen within the needle itself. The roots of lumen cleaning can be traced back to clean-in-place processes developed during the 1950s and 60s. These were applied to large industrial scale operations where it may not have been cost effective and sometimes even impossible to disassemble and clean long lengths of piping. The pioneers of these methods identified 4 key factors in a successful cleaning program: time, temperature, chemistry, and power. Time and temperature are fairly simple concepts, but chemistry and power might be a little more complex. Chemistry refers to the detergents, disinfectants, and rinsing solutions, of which there are many, that are used during the process. Power refers to the shear force that is applied through hydraulic action of the moving fluids against any contamination within the lumen itself. The right combination of these 4 factors can really make or break a reprocessing protocol and must be chosen carefully. When developing a lumen-specific protocol, we begin by characterizing the lumen as we discussed earlier, measuring it, analyzing its composition, and so on. Another one of the starting points is to classify it as a macro or a micro-lumen. In our world, macro-lumens are 0.32 in diameter or larger. This size accommodates brushes, pull-throughs, guide wires, and other cleaning instruments, as well as borescopes for inspection purposes. Now, these lumens can be cleaned differently than micro-lumens, which require a different approach that is purely chemical and hydraulic. Central scale programs in hospitals and even some other reprocessors often only use one of the 3 common methods to deliver solutions through instruments, which are injection, spring, and pumping. At Innovative Health, we use all methods when developing our protocols to ensure that our approach is both effective and efficient. So it's important that we thoroughly characterize the lumen because we need to ensure that we establish the right flow profile. Small diameter lumens like to maintain a laminar flow, but this is absolutely terrible for cleaning. If you've ever taken a fluid dynamics course, you'll recall that the flow velocity near the lumen wall is essentially 0. So any contaminants residing there, which is where they are, remain there for a long period of time, making for a very ineffective process that may not work at all. Establishing turbulent flow through the lumen allows for higher shear stress with no dead zones and greater mixing of solutions with the contents of the lumen. This removes contaminants much more efficiently. Reynolds number calculations help us determine whether our flow should be in the laminar or turbulent range, given the velocity and characteristic length. This looks fairly elementary, but it gets more and more involved as different geometries, junctions, elbows, and other features are considered. This is where simulation using computational fluid dynamics or CFD software really helps visualize flow through a lumen. We can look for eddies, backflows, and other dead zones that would prohibit efficient reprocessing. The simple solution would just be to increase the velocity of solutions moving through the device. But as we can see, as velocity increases, so does pressure. So you might achieve turbulent flow, but you'll put unnecessary stress on the device, which can affect its performance post-reprocessing. Also, move too quickly, and you'll begin to impact the chemical action of your solutions themselves. Now this really comes into play when using enzymatic cleaning agents. As we all learned in Chemistry 101, the rate of your reaction is dependent on several factors like temperature, pH, concentration, and so forth. Enzymatic detergents all have an optimal performance range, which we have to take into consideration for each particular device. Pump too quickly or apply too much ultrasonic energy, and you can actually heat your solution, which could inactivate them. Additionally, most reusable equipment reprocessing calls for the use of harsh, caustic, or other ionic solutions that are really incompatible with the materials used in the design of EP catheters. This is why it's crucial that reprocessing methods are carefully designed and tailored. Any questions so far? All right, well, now that we've all had a refresher on the first 2 years of engineering school, let's see those concepts in action. The basic reprocessing protocol involves manual removal of contamination, then pumping, cleaning, and disinfecting solutions and rinsing with pure water. Finally, a custom sterilization cycle involves deeper vacuums and greater positive pressures to ensure that ethylene oxide reaches even the most internal areas of the lumen. Let's take a look at this process through a video. Highlights the Agilis introducer. This begins to show you what we do with the reprocessing of the Agilis introducer dilator. You can see that manual decontamination is performed at the beginning. As we mentioned, we disassemble the devices, allowing us access to certain areas of the be inaccessible. You'll notice that we reprocess the introducer and the dilator separately. Besides lumens required processing parameters. Here we show the cleaning solution being pumped through the device itself, providing that hydraulic power earlier. Additionally, we reprocess the valves separately and inspect them individually as well. So as we mentioned before, with macro-lumen devices like the Agilis, we really use a two-prong approach to cleaning involving both mechanic and hydraulics. But while cleaning and disinfection are very important, they're not the entire story. I'll give you one more opportunity for some questions before we move on. So let's talk about what happens after the cleaning and disinfection process is completed. So first off, what is an occlusion? An occlusion is anything, be it contamination, material breakdown, or other foreign object residing within a lumen that restricts flow. It's important that these are detected because they could lead to device failure or increased patient risk if left behind after reprocessing. The images you see here are reusable devices, endoscopes, that went through a central sterile process and were inspected with a borescope afterward. You can see the contamination remaining on the wall and at the restriction point at a junction. Now, these are perfect examples of processes that weren't optimized for a particular device. They're also good examples of the types of contamination that we would reject for if our devices contained anything similar. Speaking of rejecting devices, a significant number of devices do not pass our strict and very conservative requirements. We reject a considerable number of devices that we reprocess for any damage, leaks, or other issues that could lead to performance and safety concerns. It's also critical that devices retain their shape. ensuring proper spacing and arrangement of electrodes, which has a large impact on mapping capabilities. Of course, you already know that we thoroughly test every single device in our process for proper electrical, location, and ultrasound performance whenever it's applicable to that device. But what about things that you can't see? Our processes must meet conservative FDA Tier 30 cleaning endpoints, along with sterility, biocompatibility, and other testing. But previously, there was no guidance on including particulates found within a lumen. Prevailing thought was that these would be discovered through the many tests assessing residual levels of hemoglobin, protein, or total organic carbon. And it is accepted that passing these tests proves the process fully removes such contaminants. But at Innovative Health, we also understood the need to identify inorganic particulates. So inorganic contaminants and particulates are often caused by material breakdown, like skiving during a transeptal needle use, or salts, contrast dyes, or other injectables that dry post-use. And any such particulate presents an embolic risk. As such, we needed to develop methods to identify and reject devices that contain even a single occluding particulate. But just like we have different cleaning methods for each device, we have different occlusion inspection methods. The principle behind these tests is that an occlusion impedes flow through a lumen, which can be measured. Considering the Hagen-Poiseuille equation, we can see that flow is a function of several factors, including the device lumen, diameter, and length. This change is based on restrictions caused by the occlusion, similar to that created by a venturi or orifice plate, which anyone with an engineering background has come familiar to. So the patent pending tests I developed are the only approved micro-lumen inspection methods available. They have become the new standard in occlusion detection. To summarize, these tests are capable of identifying a single subvisible particle within a lumen and rejecting any device that contains it. Now, don't forget, we perform this on every single device that goes through our process. Each device requires a unique test method, and to date, I have developed tests for 6 different families of devices. There are 3 devices that we are currently approved to reprocess, and next week, we will be discussing the 3 new devices that we hope to be launching soon. With that, I'll pass this to Leston.
Quickly, guys, remember to put yourself on mute if you're not presenting or asking questions. Thank you.
Yes. So, as you saw and heard, significant amount of engineering resources, development, and effort went into developing a safe and effective process to clean macro lumens of the products in our portfolio. All this is done to ensure a safe and effective product for our customers and subsequently the patients in need. Currently, Innovative Health is cleared to offer reprocessed Pentere, manufactured by Biosense Webster, a Agilis introducer sheet kit manufactured by Abbott Medical, and Volcano IVUS catheter that is manufactured by Philips Healthcare. With the patent pending method in hand, as Aaron was detailing, Innovative Health now can adapt this process to technically any lumen devices, regardless of it being in a micro or macro lumen category. As it was mentioned before by Lars and Rafael, devices are getting more complicated. So we at Innovative Health do not look at these lumen devices as a hindrance to reprocessing, but welcome this innovation as it adds significant benefits to the patient care and the clinical outcome. So this is the approach we take with any technological advancements in the field. Reiterating what Aaron mentioned, when we design the process to clean and disinfect these lumen devices, we also make sure that 1, it is clean to ensure safety, and also 2, that it still maintains the functionality and performance that's intended by the original manufacturer, but mainly to ensure the safety and effectiveness of that said device. So these, we take it as our design input, and then we turn that into our design outputs, which is the performance and the usage of the device. These requirements are extremely conservative as we understand the risk to the patients, and we hope the details on the challenge, the theoretical and practical applications discussed here was invigorating and revealing for you. That being said, this is just one of many competencies that we have. I'll let Rafal go into the details there.
Yeah, thanks, Blesson. So in closing, I think we realize the technology in the EP lab is advancing very quickly. We spend a lot of time in clinical cases. We're staying in tune with the clinical direction, the clinical disease state, where things are going in the EP world, and also all the new products that are being launched by the big companies. So we see that evolution happening very quickly. We see the product life cycle on these products shortening with every new launch. So these are all things that we have to really be aware of and understand, because again, as we pursue these technologies, we have to first develop them, apply them, clear them, and then launch new products using them. So we have to spend a lot of time making sure that we're caught up on all that. So it was really important for Innovative Health to make sure that we're focusing on all the different skill sets that are that we see coming down the pipe with new products. Today was obviously very heavily focused on the lumens. But if you look at the slide, there's a lot of other things that, things like the hydrophilic coatings on devices, component replacements. We see a big movement in the sensor-enabled devices, and us developing some of the technologies to work around that. So all these different things we're working on actively here as we partner with hospitals to stay ahead of that curve of new products that are being launched by the manufacturers. We're really excited about next week's webinar. I think next week we'll be able to show some additional things that we've been working on, some of the things that we have planned later in the year. We want to make sure that as we develop these new technical capabilities, that we are transforming those into new products to, again, make sure that we're keeping up with that technology curve as a specialty reprocessor.
Very nice. Thank you, Rafal, Blesson, and Aaron. Are there any questions? Since we now have three of the best engineers in the medical technology space in front of us. Okay, doesn't seem like it. Any last words, Aaron, Rafal, or Blesson?
Yeah, we appreciate the time. We welcome any questions. I think our focus is to be able to build confidence with our clinical partners in what we do. Certainly, we want to lead the way with new products, both current and things that are coming in the future. But we also want to make sure that we are educating everybody about what we do and how we do things. I think that's important to know that we are partnered with FDA and things that are out there as far as standards and all the compliance that we go through to make sure these products are safe. So any questions that ever come up, feel free to reach out to us.
Our contact info is available online, so we can also discuss any of this offline if you'd like to have some discussion after you get a hold of us.
Yeah, one thing I tell the physician during clinical trial is help us help you, help us get better. Because that way you guys, when you're using in the clinical setting, you get the best advice from us. When we get feedback from the physicians, we sit down as an engineering team, we go through the effort of understanding where that's coming from and what we can do to improve in our process to make that product better in the physician's hand and for the patient. So it's a feedback loop that we continuously try to make ourselves better, and that's why we as Innovative are different. So yeah, please have the questions coming, and we'll do our best to get that answers to you.
Well, thank you very much to the Innovative Health Engineering team and to everybody else for participating today. Again, next session is on Tuesday. It is on new savings opportunities in the EP lab, and we're hoping that many of you will be able to join that.
Thanks, everyone.
Thank you.